In this video, we're going to explore what happens with neurons in synapses. So basically how nerve impulses and messages get sent throughout our body. So some reactions are reflex or considered reflexes. So this is the reflex arc. So basically, if you receive a stimulus, like you've stepped on a tack or you've put your finger over a hot flame, a sensory neuron will send the signal to your spinal cord. Your interneuron is sometimes called the association neuron, will send that information directly to your motor neuron, which is a neuron that connects to your muscle and tells your muscle to get the hand away from the tack or the flame. So it's a very quick response. It's very reflex. You don't send the information to your brain to process and say, oh, this is hot. I should probably move my hand out of the flame. Or this is sharp. I should take my finger away from this sharp tack. So reflex arcs are very quick. So what are neurons? So neurons are the basic functional unit of the nervous system, not to be confused by nephrons, the basic functional unit of the kidney. Neurons carry information through the nervous system in the form of nerve impulses. And as I mentioned before, there are three types. There's a sensory neuron, which collects information and sends it to the central nervous system. The interneuron sends information within the central nervous system. And then the motor neuron sends information from the central nervous system to a muscle. And bundles of neurons are called nerves. So, to differentiate between central nervous system and peripheral, PNS, peripheral nervous system, the central nervous system is your brain and your spinal cord, and then the peripheral nervous system are any of the nerves and neurons that extend from your central nervous system to the rest of your body. So here's a diagram of a neuron. So this here, the thing that kind of looks like a star or a starfish, that's your cell body. Sometimes it's called the soma. The nucleus is found there. And then these little branches here that look like tree branches are called dendrites. They're the receiver. They receive signals. And then part of the cell body extends into this structure called an axon. So that's the conducting fiber. So this long projection, the yellow part, is the conducting fiber. It's wrapped in something called the myelin sheath, which is an insulating fatty layer that speeds up transmission. The myelin sheath is made up of Schwann cells, and they're not continuous. You can see that there's gaps in between right here, and these gaps are called the nodes of Ranvier or nodes of Ranvier. And nerve impulses used to be thought that they just traveled along the axon. We now understand that they travel by this process called saltatory conduction. They leap from node of Ranvier to node to Ranvier all the way through, and that, for some reason, causes a much faster transmission rate. The very end of the axon here, it branches off again into these axon terminals. Those are the transmitters. They send the information on to the next neuron or onto a muscle or whatever its function it's acting on. So the parts of the neuron. So the soma is the cell body. It contains the nucleus. The dendrites, we call them the receivers. They're the branches that collect the nerve impulses and send it towards the cell body. The axon carries the nerve impulses away from the cell body towards the next neuron or muscle fiber, if it's attaching to a muscle, if it's a motor neuron. The myelin sheath is that fatty insulation made of Schwann cells, which speeds up nerve transmission. The nodes of Ranvier are the gaps in the myelin sheath that allow the transmission through saltatory conduction. So that's the way that the nerve impulse skips from one node to the next as it moves down the axon. So this diagram from Cognity compares continuous conduction versus saltatory conduction. And you can see that over time, with continuous transmission, you've only moved this far, whereas it's almost at the end of the neuron in saltatory conduction. So nerve impulses are electrochemical, and the movement of ions or charged chemicals sets up an electrical current, And the membrane of nerve cells is semi-permeable. So what it does is it lets some ions in and some ions out. And that causes a voltage difference across the membrane. So resting potential. When the cell is not sending a signal, it's said to be at rest, but it's actually not just sitting there. It's actually maintaining the resting potential through something called the sodium-potassium pump. So at rest, the inside of the cell is negatively charged relative to the outside of the cell. So the sodium-potassium pump, it uses ATP to move sodium ions out of the cell and potassium ions in. But it doesn't do it at the same rate. So what it does is it pumps 3 sodium ions out and moves 2 potassium ions in. So the fluid outside the cell now contains more positive ions than inside. So the inside is negatively charged relative to the outside. And the resting potential of a cell is negative 70 millivolts. So we can see here, there's the sodium-potassium pump. It pumps out 3 sodiums and pumps in 2 potassiums. There are also 2 other channels that play a role in the action potential when we're actually sending a signal down the axon. So we'll talk about the sodium and the potassium channels later. So the action potential. So the cell surface membrane contains voltage-gated sodium-potassium channels, which are sensitive to potential difference. When the neuron is at rest, the channels are closed. The action potential happens because of an exchange of ions across the membrane. So what happens is a stimulus comes, it causes the opening of some sodium channels, causing some of the sodium to rush into the cell, and then that leads to more steps along the way. So we're going to take a look at that. So the action potential actually is set up in 3 stages. So the first is there's the resting potential. Then there's a stimulus that causes depolarization, and that's when the sodium channels open and the sodium ions move into the cell. Then they start to close and the potassium channels open, and the potassium start moving out. That causes repolarization, but it takes a little bit longer than to close. So you actually overshoot the resting potential. And so then you have to restore that back with the sodium-potassium pump. So there's a depolarization, a repolarization, an overshoot, and the resting potential is restored. We also have to reach threshold, and we'll talk about that too. So we're receiving stimuli all the time, but not every stimulus leads to an action potential. So sometimes not enough of the stimulus has occurred to cause enough depolarization to send a signal. So, depolarization. The stimulus causes some sodium channels to open. Sodium ions start rushing into the cell, following their concentration gradient. So remember, there were more outside than inside. And then the nerve cell becomes depolarized as the positive ions start surging in. So it goes from negative 70 and will rise quite dramatically. However, as I mentioned, threshold has to be reached. So it will not fire unless the potential difference reaches the threshold of negative 55 millivolts. So there has to be enough of a stimulus for that to happen. Then, as depolarization happens, that nerve impulse travels along the axon by depolarizing the adjacent sections of the axon. Repolarization is when, after the sodium channels have opened and the sodium's rushed in, the potassium ion channels open to compensate for that influx, and then the potassium ions rush out as they follow their concentration gradient, and then the sodium channels begin to close, the cell repolarizes, and the potential difference extends slightly beyond the resting potential. So that's called hyperpolarization or overshoot. The potassium channels will close, the cell will return to the resting potential through the sodium-potassium pump, and then the nerve cell can't fire again until the resting potential reaches negative 70 millivolts again, and that's called the refractory period. So you can see here, as you're moving along the axon, This one's back at rest. The signals already happened, so the sodium channels are closed. This is the refractory period. So the sodium channels are inactive because we haven't returned back to resting potential. This is the part that's being depolarized. So this is where the cell signal is happening. The sodium channels are open. And then the next region is where the next action potential will be triggered. And then so on. It'll keep moving along. So this refractory period also prevents the nerve impulse from going backwards. It only goes one direction. So once the nerve impulse has reached the end of the axon, it reaches the synapse. So nerve cells don't touch. They also don't touch muscle fibers. Instead, there's a gap called the synapse. So information travels from neuron to neuron or neuron to muscle fiber through the synapse. And it's the small space between nerve cells. And it's made of three components. The presynaptic terminal that contains vesicles or vesicles carrying neurotransmitters. It also has mitochondria and other cell organelles. So that's in the first neuron. It also has a postsynaptic terminal that contains receptor sites on the dendrites for neurotransmitters. And that's in neuron 2. And then there's the cleft, the synaptic cleft, or the space that's between the two neurons. So let's have a look at that. So here is our presynaptic neuron. This is our postsynaptic neuron. This is a vesicle or vesicle with neurotransmitter. There's the receptors all along here. This is the cleft, the space in between. So yes. So this is what happens. So an electrical impulse travels down the axon to the synaptic terminal. It causes it to get depolarized. In this case now, calcium channels open and calcium ions enter the neuron. The influx of calcium ions cause vesicles containing the neurotransmitter, like acetylcholine as an example, to migrate into the presynaptic membrane. The vesicles will fuse with the membrane and release the neurotransmitter into the cleft, the space between. The neurotransmitters will go across the space and fuse to the receptors for that neurotransmitter on the postsynaptic membrane. And that will cause a nerve impulse to continue down the next axon, or if it's at a muscle fiber, it'll cause the muscle fiber to react. So some common neurotransmitters. Acetylcholine, which is an excitatory neurotransmitter. It's the one that's involved in central nervous system nerve transmission. It acts on striated muscle fibers and causes your muscles to contract. The enzyme acetylcholine esterase breaks down the neurotransmitter into acetate and choline so that they can be released in so the muscle can relax. Another one is gamma aminobutyric acid, or GABA, which is an inhibitory neurotransmitter. And it calms and reduces the activity of neurons. So many anti-anxiety medications actually work to increase the amount of GABA in synapses. So it reduces the amount of signaling. So it calms and reduces the activity of the neurons. So neonicotinoids. These are derivatives of nicotine that are used as pesticides. And they're actually used as natural pesticides. But they can irreversibly bind to synapses. And these synapses are the ones where acetylcholine would normally bind. So that means that acetylcholine esterase can't break them down, and it prevents nerve signal propagation and involves in paralysis and even death of the insect. But we have to be careful because they can be harmful to us as well. So while these pesticides aren't directly harmful to humans, they do affect us through the food chain. And they're leading to a decrease in honeybee populations, which are a major pollinator. And birds who eat these insects are also being affected due to the loss of their own food sources.